Antibodies are great at targeting protein markers on cell surfaces, and tagging those cells for destruction. This would make them very suitable for therapeutic use in diseases such as cancer, where they could single out tumour cells for the immune system to attack. One major issue is that most of these antibodies are generated in mice. If mouse antibodies are given to patients, the immune system would flag them up and clear them out. Scientists have found 3 ways to get around this problem. Because the substrate recognition only happens in the antibody's variable domains, you can take the mouse antibody's variable regions and graft them onto human constant regions. These are called chimeric antibodies. A second approach is to replace only the residues in the CDRs at the tips of the variable domains. This leads to human antibodies that mimic the exact target binding characteristics of the original mouse antibodies. These are called humanized antibodies. And thirdly, fully human antibodies produced by transgenic mice can also be used for therapeutic purposes. Chimeric, humanized, and human-derived antibodies are being used to successfully treat cancer patients, for instance in the case of HER2-positive breast cancer. Healthy cells have low numbers of HER2 receptors on their cell surface. These HER2 receptors occasionally transmit a signal, telling the cells to divide. In the case of HER2-positive breast cancer, the number of HER2 receptors on the cell surface is increased by up to 1,000-fold. This HER2 overexpression leads to far too many growth signals, causing cells to divide uncontrollably. Therapeutic antibodies against HER2, such as trastuzumab, a humanized monoclonal antibody, are routinely used in the clinic to treat HER2-positive breast cancer. They bind to the extracellular domain of HER2 and attract the immune system's natural killer cells. Fc receptors on the NK cells recognize the therapeutic antibody and release cytokines that force the cancer cell to self-destruct. Using chimeric, humanized, or human-derived antibodies can successfully target particular types of cancer at a molecular level, which has proven to be a major therapeutic breakthrough.